Okay, welcome to lecture 5.2 on DNA replication. Today we're going to be talking about how DNA goes from being a double-stranded, double helix into two identical copies of each other. It's actually kind of important to remember that this happens during S phase of mitosis, or not mitosis, but of the cell cycle in general. So S phase of the cell cycle. Since we've already talked about this, it's a good idea to keep the big picture, right? So we've got DNA replication that's happening in S phase, and remember the goal of DNA replication was to create copies of DNA. So that's what we're going to look at today and see how it is done. Okay, first we're going to look at DNA. DNA is in every organism. Remember, DNA was a characteristic of life. The fact that an organism had DNA was a characteristic of life. So in prokaryotes, the DNA is found in the cytoplasm because remember, in prokaryotes, there's no nucleus. But in eukaryotes, it is found in the nucleus, okay? Also, in bacteria and prokaryotes, it consists of a singular circle piece of DNA, whereas in eukaryotes, like you have 1,000 times more DNA, and they exist in the form of chromosomes, which looks like this. When we go through S phase, or replication, we turn that simple chromosome into a duplicated chromosome, which is what we're going to look at today and see how that is done. All right, so this is DNA replication. Replication, again, remember, means that we're copying the DNA itself, so we're copying the DNA, and that way when the cell duplicates, each cell has an exact copy. So how is this done? Well, the DNA separates into two strands. So here's my first strand, and we're separating into one here and one there. And then as they separate, new nucleotides form another side of the DNA, and that's that red piece you can see right here. And they match up to the already present DNA strand that's unwinding. So what you actually end up with is one strand into two strands here by two strands. So let's look at that from another view in a second here. So just to summarize, the two strands unzip, and when they unzip, each strand acts as a template or I would say a base for the other strand to form off of, okay, to form a complementary strand. So if we have our strand here, for example, here's my template, my copy or my complementary strand would match up, right? So A with T, T with A, C with G, G with C, going all the way through. Every time you split the DNA, you make an exact copy of it through replication, and these nucleotides match up just the same as they started out in the original strand. So the result is then two DNA molecules that are identical to each other, completely the same. All right. Here's another way of looking at it. Here's DNA replication again. We've got our original strand right here that's splitting off into two. Think of it as kind of almost like a zipper unzipping. Okay? If you picture it that way, or you have a coat on right now, or a jacket, you can do that. That's exactly what's happening. The strands are unzipping, and then nucleotides or extra letters, right, are filling in the open nucleotides pieces. So if you unzip your zipper, you've got open prongs now showing. Think of it almost like you would unzip your zipper, and then a whole half of the zipper would just come in and fill in those gaps. So you would just have two zippers. Your jacket would be ruined at that point, but that's kind of what's happening here. We're starting with one strand, we're going to two, and the strands are unzipping to get that done. Okay? The main enzyme that is involved in this is called DNA polymerase. We're making a polymer. Right? Making a polymer of DNA. So this is the enzyme that does that, DNA polymerase. The enzyme is involved in replication, and it helps form new DNA. It also proofreads the DNA and looks for mistakes. If any mistakes are made in the process, it will fix those too. There's one other enzyme I kind of want you to know about as well, and that is helicase. Helicase unwinds double helix. So there are many different enzymes involved in this process, but here are the two main ones. One enzyme involved in unwinding the DNA, and the other enzyme involved in making a new piece of DNA. Okay, so that's it for DNA replication. If you have any questions, feel free to ask in class. And again, we'll have our quiz on this coming up shortly, so keep studying and reviewing. All right, great. See you next time. Bye.